So thank you very much, everybody who's been listening so attentively. And I want to give you a little bit on the research and to tell you that, first of all, before you understand the research, there's such a hype now with COVID and clinical trials. Everybody has got misconceptions about clinical trials. And I want to tell you that clinical trials are human studies designed to answer specific questions about safety and effectiveness of drugs and other interventions. They are registered and they're controlled, and they're usually in 3 phases. So if you're a technician or a geneticist working in a laboratory and you have a bright idea about something that could possibly be of benefit or good new treatment, you would go into the laboratory and look at that, either the genetics or the biochemistry, and investigate that particular intervention. And if it looks promising, you will then go to a cellular model. Now, stem cells, one of the benefits of stem cells is now allowing us to grow diseases, specific diseases, in addition. Again, if you have good success, you can then go to an animal model. That's usually a mouse model, can be a naturally occurring mouse model with retinal degeneration, which does happen, or it can be a transgenic mouse or animal where the genetic mutation is actually read into the animal. And then that is what we call preclinical studies. Now, if that looks really promising, then you of course have to find a funder, and you put together a clinical trial protocol. You have to specify exactly what you're going to do, what the outcomes measurement is going to be, how can you judge the success of that trial, what is your treatment, the levels of dosage, etc. You'll go to a trial, like the FDA or the European Union, or in South Africa, you go to the clinical trials regulatory body, present your protocols, and hopefully they will give you permission to go to a phase 1 clinical trial, which is very few people. And of course, especially in something that like gene therapy or stem cells, which is quite novel and quite invasive, these people will have to stay close to the clinic so that they can be checked regularly. Now, if that phase 1 clinical trial is successful, usually takes 2 years, you go to the authority, present your data, and hopefully they will give you permission to go to a phase 2 clinical trial, which will be more people, and you sometimes will even then go to multi-centers if they're orphan diseases or rare diseases and you don't have enough patients on your own, close to your own laboratory. And then again, after 2 years or so, and your phase 2 clinical trial is successful, you'll take your results to the regulatory board, and you will present them with your results, and hopefully they will give you permission to go to a phase 3 clinical trial, which is many more patients, and those will definitely probably have to go to multi-centers. So that's your phase 3 clinical trial. Again, at the end of possibly 2 years, you take your results to the regulators, you prove your results, and with all things being equal, you will be able to have your intervention registered, and you'll be able to sell it to lots of happy people and restore vision. So that is how a clinical trial is. I'm not going to spend too long. Hazel showed you very well how the eye works, and you know, of course, that the eye has to focus on the retina at the back of the eye. And Hazel told you about rods and cones, so I don't have to spend time on that. And this is just such a cool picture from Professor Joe Holyfield of what the retina would look like if it was depicted almost like a multi-layered sandwich. From the back of the eye, the white thick covering, the sclera. Hazel told you about the choroid, the blood supply, and the chora capillaris. And then we get Bruch's membrane, which we're not going to talk about today. And then the pigmented epithelium layer that Hazel mentioned, which is the nurse layer and the factory layer. And then you get the photoreceptor cells and the rods and cones. And then you get this neural layer, and that's the fovea that Hazel depicted, where the cones are in such high concentration. So I just want to show you an artist impression of what the retina actually looks like. We'll track the journey of light as it comes from the vitreous through the optic nerve, and then it will go through the neural layer. So we'll come back to those cones and then those cells, and then it comes into contact with the photoreceptors, the rods, and the cones, although the cones are depicted here as color red, green, and blue. Of course, they're not that color. That's the wavelengths of light that they respond to. So the light does now come through, and it has reacted with the cones, the rhodopsin in the rods, or the cone opsin in the cones. And then a very complex and very not completely understood biochemical reaction starts to happen. This light impulse is now being changed to a neural impulse, and it goes through the horizontal cells, the bipolar cells, the amacrine cells, being changed all the time with these biochemical reactions till it comes into contact with the ganglion cells. Now the tails, or the axons of the ganglion cells, are long neural fibers, and they join together to become the optic nerve, and that changed message is then sent to the brain, where a whole other series of changes take place, and we're not even going to talk about those today. I want to just tell you that there's two important things I need to tell you. Firstly, about the neural layer, that as these cone and rod photoreceptors are dying or becoming dysfunctional, this neural layer remains pretty intact, and that will be important later. And also that, as Hazel spoke to you about the pigment epithelium layer, particularly in Stargardt's and in dry AMD, the initial problem is not that the cone photoreceptors die, is that you get waste products being built up in the pigment epithelium, and the cone death is secondary to that. So what causes genetic retinal conditions? Well, there are genetic mutations, mistakes, that produce a toxic or missing retinal proteins, and these cause the death or inactivity of the photoreceptors and vision loss. They can also cause misfolded proteins or waste products that clog the cells. There are more than 300 genes that already have been implicated in retinal degeneration, and they can be expressed in any cell of the retina. Some gene mutations or mistakes are found in the RPE, the photoreceptors, the neural cells, or the choroid. Because the retina is one of the most metabolically active sites in the body, it doesn't respond well to any information that is fed to it that is incorrect. And then the resultant end product, of course, is the death of the photoreceptors. And so far, only one treatment has been registered, and that's gene therapy. We will come back to that. So these are the potential therapy pathways. Gene therapy to either replace a gene or send a missing copy of the gene to the cell, or to deliver DNA that will produce a therapeutic substance. We then have stem cells, which they're using to grow new photoreceptors and also to grow new retinal pigment epithelium. We then have gene editing, which is like a cut and paste type of mechanism that you use on your computer. Gene editing uses a cut and paste in the DNA. You can have messenger RNA editing or CRISPR Cas9. Then you get Optobionics, which are photoswitches. And then you get a whole basket full of pharmaceutical interventions. And things like anti-VEGF that Tracy's described so well comes into that basket. We also get visual cycle disruptors, supplements of pharmaceuticals, proteomics, and there's a whole range of different pharmaceutical interventions. Let's look more closely at gene therapy. If you've got Stargardt's or a recessive RP, for example, you inherited a malfunctioning gene from both parents, so you have two copies of a gene that has the wrong message. And gene therapy can introduce a working copy of the gene to the retina. It can be it's delivered by a viral vector, an adeno-associated virus, or a less popular or less effective lentivirus, or you can use a viral vector to deliver a gene that will produce a therapeutic substance, and they're using that in photoswitches. I've shown you this picture of adeno-associated virus because everybody's seen what the coronavirus looks like. And I want to assure you that the AV is a very different virus. As you can see, it's a very safe virus and it's a very easy virus for the researchers to work with. Every living organism has DNA in the cell. So what the researchers do is they take this AV and they remove the DNA from the virus, and into that they insert the copy of the DNA, the basic genetic code that is missing in the eye, and they insert it into the virus, and then they replicate the virus. The viruses replicate easily. They make thousands of copies of the virus containing new genetic material, and they use a syringe to deliver those new code in the virus to the pigment epithelium. And I think it's miraculous. I think that people don't realize just how wonderful this is, that these cells in the pigment epithelium actually pick up on this new genetic information and say, hey guys, look, we've got a new genetic code, let's try it. And they do, and it works. So let's look at this gene therapy. It's the first ever gene therapy for an ocular disease. It was approved by the FDA and EU in 2017. It's called Luxturna, and it treats the RPE65 mutation in LCA and early onset RP. It was brought to market by Spark, and it's being licensed by Novartis in Europe. The clinical trial began in 2007, so it took 10 years. The good news is it works. The bad news is the cost, and we won't even go there. So let's look at stem cells. Stem cells are the pluripotent early embryonic cells. They're like Superman cells. They can and do become any cell in the body. They change and morph as the embryo grows. We also have induced pluripotent stem cells. They are induced from other body tissues like blood or skin or bone marrow. And researchers are growing photoreceptor cells and pigment epithelium cells from both types of stem cells. I'm not going to spend any time on dry. Hazel and Tracy both covered this well, and you know the difference between dry and wet, that there's no treatment for dry, and for wet there are anti-VEGF treatments, and they don't actually know what actually causes. We know it's multifactorial. Let's look at photoswitches, which is a very exciting new type line of research. We said those neural cells are pretty intact, and the researchers said, hang on, why are we trying to develop an artificial retina when we've got a layer of cells that are still working? They're not light sensitive, but that's not such a big problem. Let's make them light sensitive. And they're doing that with channel rhodopsin, which is a naturally occurring photosensitive molecule, and it's being genetically delivered to ganglion cells, and there are patient trials, and the new trial is very exciting. Professor Sheila Nirenberg said 5 years ago, we need to decode the message. It's not just changing the light to a neural impulse. There is a language of the retina that we don't understand. And she spent the last 5 years decoding that language. And she now has bionic sight, and they've treated the first patient with gene therapy and with glasses. So the gene therapy delivers a molecule to the ganglion cells to make it receptive to this special code that is generated by a device that is in a pair of sunglasses. And then the message that's being sent to the brain will be in a code that the brain understands. And unfortunately, there's only one pair of glasses so far, and they're staying as safe in an office. Otherwise, I'd be there like a shot. So let's look at gene editing. Harvard has said they can now edit out a single DNA letter. So genes are made up of 4-letter words containing 4 letters, And 3 of those letters form a little codon, and they CGAT. And now instead of moving out the whole gene, they can actually just remove one of those letters. And very often in a genetic mutation or a genetic condition, it's only one of those letters that are wrong. Then we also get messenger RNA editing, which is antisense oligonucleotides, and you all have to tell me that at the end of the talk, what exactly that is. And ProQR are in a phase 1 clinical trial for Ushers and for LCA10. And what this does, it actually masks the mutation so the cell can read over that piece. It's like if you have a sentence, the cat sat on the mat, and you remove sat, the cat on the mat, you'll actually put, your brain would put the missing words in, or here, the cellular structure will actually put the missing DNA in. And CRISPR Cas9 is a great tool for editing. It's a natural phenomenon that bacteria have been using to disable viruses probably since the dawn of time. But the mechanism was only discovered 5 years ago by Elizabeth Doudna, and it's being extensively used in cancer and human genome. It has been used to edit genes in embryos, but they're not allowed to be implanted because there is an international moratorium on editing human genome, human embryos, because that means we can actually change the germline. But there's now a phase 1 clinical trial called Brilliance that KCI Institute are using for LCA10, and it's just begun. So that's very exciting news. Pharmaceutical trials where we're looking at improving anti-VEGF for AMD, and there's now a new implantable device called a port delivery system, which they're going to implant. It's the size of a grain of rice into the vitreous, and that will be filled with anti-VEGF, and it can be topped up in the doctor's office. Drugs to slow the waste product accumulation, we'll come back to that. Antioxidants, and they're also looking at repurposing of existing drugs, and they're spending a lot of money on that. So what does Retina South Africa do? We identify patients, we inform, educate, counsel, support, we do intervention where we need to. James has told you all about apps and assistive devices, and we also will refer you to service providers. We run a very comprehensive, confidential patient registry. We facilitate genetic testing. We collaborate with our clinicians, the Division of Human Genetics at UCT, and international researchers, because our determination is to bring clinical trials and accessible treatments to all South Africans. So I'm often asked, how do I get onto a clinical trial? Well, first remember that a clinical trial is not a treatment. Most trials are masked in that half people will get a placebo. You do need a genetic diagnosis, even if it's not a gene therapy. They need to make sure that they're treating the right patients. If you have a rare disease, you will need to recruit your family members, because you can't go overseas and join a phase 1 clinical trial. As I said, you need to stay close to the clinic. So our hope of getting you onto a clinical trial is to bring clinical trials to South Africa, because you need that access to the clinic. And we now already know that for multi-center trials where they've got rare diseases, they're going to need more patients than they've got access. So for phase 2 or 3, we could be included. But patients know they need to comply with all the inclusion criteria. So if you're interested in clinical trials, you can look at clinicaltrials.gov, and there are thousands then. I'm not going to go through them. But remember, they're not all FDA approved. So don't fall for something that's listed on the clinicaltrials.gov. Be told, yes, you can come onto this and pay and be included on a clinical trial. It's not a clinical trial. A far better, reliable source of clinical trials that are in the pipeline is the American Foundation Fighting Blindness.org. And if you go and look at their clinical trial pipeline, you'll see there are 38 conditions in different types of clinical trials: gene therapy, optobionics, cell-based therapies, or molecules, proteins, and inhibitors. So if you're interested in clinical trials, please go and look. And that's a great source for all sorts of information. Somebody asked a question about clinical trial in South Africa. Well, we were approached to recruit patients for Exustat, which is a phase 2, a double masked. So in other words, neither the patients nor the doctors know who's getting placebo. And it's a vision disrupted drug, that's the Exustat, and it's a daily tablet for 2 years. So we approached 320 of our members who had, no, I retract that. 320 of our members over the age of 16 had a clinical diagnosis for Stargardt's. That was the first criteria. We looked at those, and of those 320, 250 had a genetic diagnosis of ABCA4. That was the second criteria. We then contacted those patients and said, the next obstacle is how much vision you have. So they had to go to their optometrist and have a best corrected visual acuity test, which Hazel will explain to you what a visual acuity test is. And they had to have a certain level of vision. That was the third criteria. Then those patients who had that correct level of vision had to go and have another test, which was to see the size of the lesion. Remember, we spoke about a buildup of waste product in the pigment epithelium for Stargardt's, and that waste product are called lipofuscin, and they can actually measure them. And that is the outcomes criteria for this trial, is they have to select lesions of a certain size, and they will know where they are, and then after 2 years, during the trial and after 2 years, they will see how did the size of those lesions pan out. Did they stay the same? Did they get bigger, or did they get less? And that is going to be very interesting. And we hope, because we had 30 patients that were actually included in the trial, of the 30 participating clinics all around the world, we actually had the most patients. And this is only a phase 2, so we're hoping for a phase 3 that we will be able to participate in that. James, in fact, asked me to add this caution. Please be aware of quacks, scams, and the internet, which is the home of quacks and scams. Do not believe everything you see on the internet. Clinical trials are free. A registered clinical trial has an FDA number. It's an NCT number for FDA and an 8-digit code for the European. If it's a clinical trial, they will have that code, and anybody inviting you will be able to give you that code. If someone is selling you a medically improved treatment, please request results from their clinical trial or from peer-reviewed articles in medical journals. If any intervention has not been tested on your specific condition, and I mean gene-specific condition, assume it's not safe to use. I'm horrified that people with Stargardt's are using Ocuvite. Ocuvite is a brilliant tablet to slow the rate of conversion from dry to wet AMD. It was formulated for AMD, and it was tested on AMD. There is another one that you can use for Stargardt's. If you're having acupuncture or microcurrent stimulations, and you say, but look, my vision is improving, yes, because they found that they do produce growth factors, which may temporarily improve your vision. But even researchers working with it have said to me personally, they don't know the long-term effects of continually producing or producing growth factors in the retina. And if you're not sure, please call us. So why do you need a genetic diagnosis? Well, you need to identify the disease and manage it. If you can get the gene, you can also find exactly what type of inheritance it is. So you can look at family members who are at risk, or if you've got a double copy of a recessive condition, for example, you can have your spouse tested, and then you can have informed reproductive choices. And possibly you may be included in a therapy trial, because we know future therapy are gene-specific and even mutation specific. We must pay homage to our wonderful partners at UCT, Professor Ramazan in the center in the front, with his brilliant team of gene hunters. We could not have got this far in our search for treatments and to bring clinical trials to South Africa without them. Please protect your vision. Hazel spoke about UV light and blue light. If you've got a good low vision therapist like I have, she's actually, Hazel has actually put a protective screen for blue light so that I can wear my glasses and not cut down my electronic device time too much. As Hazel said, no smoking. We know that antioxidants do slow the rate of degeneration. Retina Plus for inherited retinal degeneration and Ocuvite for AMD. I can't stress long enough and hard enough and often enough, no vitamin A supplements for Stargardts or cone rod dystrophy or smokers. And you really do need to make sure that even if you haven't got an ABCA4 result, that you must not take vitamin A if you've got a central vision loss. You must reduce your food additives, preservatives, flavorings, artificial sweeteners, and colorants. They are nerve poisons. A healthy diet and physical fitness, Hazel's mentioned, and sleep is very important. Take-home message. Please have an accurate clinical diagnosis. I'm amazed at how many people contact us and don't know the name of their condition. You need a genetic diagnosis because the age of personalized medicine is soon going to be upon us. You must support Retina South Africa. We need you. And you must register with myretinatracker.org because that is a portal and an anonymous portal that will link researchers with people with rare retinal conditions. And how can you support us? Well, you can nominate Retina South Africa as your beneficiary at my school, and at no charge to you. Every time you swipe your card, we do get a small donation. You can support our Circle of Light. A year's subscription is only 250 Rand, and you can stand to win a monthly prize of 500 Rand. Please support us with your membership fees and donations. And if you know people or you want to give a bigger donation, you can get a tax-deductible certificate as we are registered as a PBO. You can get hold of us at our head office. Linda's very good about returning emails quickly. You can call us on our share call or you can WhatsApp. And thank you very much for your attention.
Thank you, Claudette. That was really, really so informative and so comprehensive. And every time I listen to you, I learn a tremendous amount.